Good afternoon, students. I'm back with the biology class, and today I'll discuss the chapter Biological Classification. Today is the part 1 of this chapter. See, students, since the dawn of civilization, there have been many attempts to classify living organisms. And we have seen the advantages of classification in the previous chapter. So it was very instinctive right from the primitive time to try to classify organisms. Initially, this classification was based on just the observable external characteristics. Like the first attempt to classify the organism was made by Aristotle. What he did? He classified the whole lot of organisms into plants and animals. He further classified plants into the size of the plant. According to the size of the plants, the classification was based as herbs, shrubs and trees, depending upon the size of the plant, the branching and some observable characters. The next classification of animals was based on the presence and absence of red blood cells. So on the basis of presence of red blood cells in their blood, the animals were classified into Anima having the red blood cells and Anima without the red blood cells. The first scientific classification was attempted by Carolus Linnaeus. He proposed the two-kingdom classification. In two-kingdom classification, he classified the organism into plants and animals. But as you know, this classification was very crude, very primitive time, and there were many conflicts among the characteristics of the organisms to be classified into a single group. So, in the next classification, which was proposed by Haeckel, all organisms were classified into three kingdoms, namely Plantae, Animalia and Protista. So, Protista actually included all unicellular organisms, both prokaryotic and eukaryotic. Earlier, all these were placed under Plantae. This was also insufficient because the prokaryotes were kept with the eukaryotes. So, Copeland in 1956 proposed the four-kingdom classification. He classified organisms into Plantae, Animalia, Protista, Monera, thereby separating the eukaryotes from the prokaryotes. But this also had some limitations because fungi were placed with Plantae, but they were non-chlorophyllous. As we know, the Plantae were the organisms which could photosynthesize. Fungi could not photosynthesize. So, the next level of classification was proposed by R. H. Whitaker. He proposed five-kingdom classification that you are going to read in class 11. So, he classified organisms into Monera, Protista, fungi, Plantae and Animalia. He used certain characteristics to classify these organisms, and he also kept in mind the evolutionary relationship among the organisms. So phylogeny was taken into account during the classification. So what was the basis of five-kingdom classification? The organisms were classified according to the cell type. You must be knowing there are two types of cell: prokaryotic and eukaryotic. So, all prokaryotic organisms were grouped together and eukaryotic organisms were grouped together. You must be knowing the prokaryotic organisms were the members of the group Monera, the first kingdom, and eukaryotes included Protista, fungi, Plantae, and Animalia. The next, cell number was taken into account. So according to the cell number, the organisms were classified as unicellular and multicellular. So which group was of unicellular organism? Which were eukaryotic? They were Protista. And all other were fungi, Plantae, Animalia were multicellular. So mode of nutrition is the next basis of classification. The mode of nutrition was autotrophic, and heterotrophic. So Plantae were kept under the autotrophic, multicellular, eukaryotic organism. So they were Plantae. And heterotrophic, the fungi and Animalia. Next is presence and absence of cell wall. So presence of cell wall, it was fungi. So heterotrophic mode of nutrition with cell wall were fungi, and heterotrophic mode of nutrition without cell wall were Animalia. So in this way, he classified the organism into 5 kingdoms. Later on, the 3-domain system was proposed that divides the kingdom Monera into 2 domains, the Archaea and the Eubacteria, leaving the remaining eukaryotic kingdom in the 3rd domain. Thereby, a 6th kingdom classification was proposed, where Monera were divided into Archaea and Eubacteria along with fungi, Plantae, Animalia as the 3 kingdoms. Now students, this is just a recap of what you have learnt in class 9. Now we will start with the first kingdom, that is Monera. So what are Monera? From the previous discussion, you can say Monera are the microorganisms including bacteria, which is found everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitat like hot springs, that is at very high temperature, at very salty conditions, then in deep oceans, in deserts and snow-filled areas. So they are omnipresent, they are present everywhere. Now, you must be knowing they also live in other organisms as parasites. Now students, these bacteria are classified into 2 groups. One is called archaea, bacteria, and the second one is called eubacteria. Archaea means primitive, so they were the first bacteria to originate during the origin of life on Earth. They were the first organism to evolve on Earth. So they are present in extreme conditions like very high temperature, which we call thermophiles, at very high salt conditions, that is called halophiles, at the places where they produce methane gases, which are called methanogens, and so on. They have structure entirely different from that of the eubacteria. You must have heard about the methane gas produced by the cow dung, because the cow dung contains abundant of these methanogenic bacteria. The second group is eubacteria. You know, in biology, U means true. They are the true bacteria. Now, bacteria are classified into various types depending on their shapes. So, there are very many types of classification of bacteria, but the most primitive type of classification of eubacteria is based on their shapes. These are mainly the heterotrophic bacteria I am talking about. So, on the basis of their shapes, they can be cocci, singular coccus. So, coccus bacteria are spherical in shape, like the small ping pong balls. The second is bacillus. You all have heard about lactobacillus. Yes, this bacillus, lactobacillus, is a type of bacillus bacteria, and the shape of these bacteria are rod-like or cylindrical. The third is Vibrio. Vibrio are comma-shaped bacteria. Then you have Spirillum. Spiral-shaped bacteria. And these bacteria can be present in bunches where we call them staph. So, Staphylococcus means coccus bacteria existing or appearing in bunches. And when I say they are present in chains, that is your strepto. So, streptobacillus means the bacillus which are appearing in chains, existing in chain form. And Staphylococcus means the bacteria which exist in bunches. At your age, all of you suffer from the acne infection. This acne infection is due to this particular type of bacteria, that is Staphylococcus aureus. You have heard about lactobacillus, an example of bacillus bacteria. You have heard about Vibrio cholerae, the causative organism of cholera. That is a Vibrio-shaped bacteria. And again, you have different staining techniques on which the bacteria are again classified. One very popular staining techniques of bacteria is called the Gram staining, where the entire bacterial population are classified into Gram positive and Gram negative. Here the bacteria are actually stained with a primary stain like crystal violet and then washed with acetone alcohol mixture and then stained with a secondary stain like safranin. If the bacteria retains the crystal violet color or crystal violet dye and appear violet or purple, they are called the gram-positive bacteria. And if they do not retain the crystal violet color after an acetone alcohol wash, they take the secondary stain, that is of safranin, and appear pink. They are known as the gram-negative bacteria. Now students, again these bacteria are classified into autotrophic bacteria on the mode of nutrition. On basis of mode of nutrition, they are classified into autotrophic bacteria and the heterotrophic bacteria. So autotrophic bacteria are the one which can synthesize their own food. They are the photosynthetic autotrophs and they have chlorophyll A as the pigment. Chlorophyll A as the pigment, and they are called cyanobacteria. Formerly they were known as blue-green algae. Formerly they were called blue-green algae. The cyanobacteria are unicellular, colonial or filamentous bacteria which live in fresh water or marine water or even have terrestrial habitat. The colonies are generally surrounded by gelatinous sheath and they often bloom in polluted water bodies. Some of the organisms also fix atmospheric nitrogen in specialized cells called heterocysts. So heterocysts are the specialized cells which can trap atmospheric nitrogen, which can fix atmospheric nitrogen, like nostoc and Anabaena. So the example are nostoc and Anabaena. Now, chemosynthetic autotrophic bacteria, they oxidize the inorganic substances such as the nitrates, nitrites and ammonia and use the released energy for ATP production. So they play a great role in recycling the nutrients like nitrogen, phosphorus, iron, sulfur. The next group of bacteria are the heterotrophic bacteria. Heterotrophic bacteria. They are the most abundant one in nature. The majority of them are decomposers and they have many of them have significant impact on the human bodies. You must be knowing about the lactobacillus which makes curd from milk, the production of antibiotics, the nitrogen fixation in legumes. They are all brought about by the heterotrophic bacteria, though they also act as the pathogen causing diseases like cholera, typhoid, tetanus, etc. Bacteria reproduce mainly by fission. Under unfavorable conditions, they sometimes form spores. They also show a sort of primitive sexual reproduction known as the conjugation. There's another group included in Monera which is called Mycoplasma. So what are Mycoplasma? These are the organisms that lack completely a cell wall. They are the smallest living cells and can survive without oxygen. Many mycoplasmas are pathogenic to animals and plants. So, this is about your kingdom Munera. Thank you and